Okay, we are being recorded. So now I will introduce our first speaker, Luis Palazzesi, who will present their talk on Compositae, what fossils and phylogenies can and can't tell us.
Okay, thank you, Abigail. Thanks for having me, and nice to be included in this spot with other Latinos. I'll do my best to share my screen. Sorry. So, are you seeing the complete?
Yes, now it looks good.
Yes.
It looks good. We're just seeing your slides.
Okay.
Perfect.
Oof. Well, though this is the title, as Abigail just said. I'll give you just a brief introduction about the composite fossil record through time, with a special emphasis to the Patagonian fossil record. So this colorful chart on the left is the time dimension. So if you are not usually you don't work on fossils, you'll probably be lost. So I'll give you the ages on the left. And to give a bit of a context, this is the oldest Uticode fossil record, which are pollen grains from the early Cretaceous. These beautiful records come from Europe, from the Bohemian, which is 125 million years ago. And the oldest composites come from a bit later, from the Cretaceous, from the Upper Cretaceous or Late Cretaceous, from about 70 million years ago. We assigned those records to Parnadicivity because we found common characters to that subfamily. The oldest capitulum come from Patagonia, the oldest, well, and the only one from the moment. And we assigned it to the Museo, and we also found some pollen grains attached to the same rock. And as I said, these fossils come from the Eocene, which is roughly 45 million years ago. And the onset of diversification of some South American native groups occurred during the Eocene and Oligocene. For example, Barnetticivity, Muticivity, and other members of the Muticivity also. They are not really easy to identify because they have really, really tiny spines. So they are probably, most of them are overlooked in the fossil record. Wait, sorry. And during the Oligocene and early Miocene, which is roughly 23 million years ago, there's a tremendous increase in the diversity forms of Asteraceae, especially in South America, Australia, and even Antarctica. But the oldest records come from lower latitudes from the American continent, especially from Mexico. But what the fossil record of Compositae can tell us. So fossils can tell us what living group they belong to. Usually, the younger the fossils, the more confident the taxonomic or the phylogenetic placement. There are several softwares to do that. For example, Rev-based, Beast, among others. What fossil record of composite can also tell us? Well, the time calibration. Usually we need some previous knowledge on the distribution of fossils through time, especially for patient analysis. So it's always good to have a paleobotanist collaborating with us. The fossil record can also tell us about the existence of specimens that don't share all characters with the living species. And hence we consider them as extinct. For example, well, these are two records from the Cretaceous and the Eocene with Tulipheris, Deslillet, and Muticipalis Theieriae. They don't share all characters with the living species, so we consider them as extinct. What the fossil record of composite can somehow tell us? We can reconstruct the climatic conditions using the niche of the nearest living relative, but it's important to use like more modern, like post-Miocene fossils to do that, because if you consider that the pre-Miocene fossils that belong to extinct clades, it's more difficult to reconstruct with confidence the living conditions, the relative conditions, sorry. But what the fossil record of composites cannot tell us. The analysis of the fossil record, as I said before, can tell us if specimens represent an ancestor, but living species are not ancestral. So the living species that represent those extinct species or fossil species are not early divergent or basal or fossil livings. Many fossils have been assigned to these South American groups, shown in the bottom of this tree of Panera and Crosser published in 2016. These clades or these groups are usually regarded as basal, probably because the tree is ordered or ladderized. So the least diverse groups are on one side, and the most diverse groups are on the other side. I'll briefly show you some of those species so you can have a broad idea. What am I talking about? This is, for example, Barnetiae, some examples of Barnetiae, Chuquira garnaloa, Barnetiae, to another Barnetiae species from Argentina. They're beautiful, and some of them are bird pollinated. These are some examples of the Medici and Steve JD, well, from Brazil, northern Argentina, Prostia, Monasteries. But why these are not ancestral? Let's see, for example, analogy from the Simpsons family tree, which is one of my favorite shows. So if I'm Bart, or for example, or Lisa, Ling here, who is the Selma adopted daughter, she's my cousin. Ling, she's not ancestral or early divergent or basal. She's just my cousin. Let's move to a more scientific example. I have a tree. Let's assume it's a complete tree A from Asteraceae, and red is Barnetiae, and blue is the rest of the Asteraceae. And if we assume that if we have a time machine and travel to the Eocene with T1, and I reconstruct a phylogenetic tree in the Eocene, which is B here, and we'll have 5 species of Barnetiae and 1 of the rest of Asteraceae. So which is basal, which is early divergent or ancestral here? None of them. They're just sisters or cousins. If you travel to the present time again and see, we see that many species became extinct, and at the present we have only 2 Bernardiidae and 5 Asteraceae, rest of Asteraceae. And here again, which is the ancestral or derived or whatever, advanced or modern, none of them. They're just 2 sister clades. Of course, they could have retained some ancestral characters. This is, well, some species of Gogonaptinae, or this is species, I think, this picture was taken by Mauricio, I think, the Nicothamnus. They could have retained, of course, some characters, but it's misleading if we think that consider them as ancestral, because we tend to think that evolution has stopped for them, and no changes occurred since their early evolution, since the family originated. And it's kind of ironic because many of the species of those tribes are morphological and physiological adaptations to extreme climatic conditions, conditions that didn't exist in the past. For example, we have with the hammer, a Chuquiraga member of the Bernardiidae, which grows in Patagonia under extreme climatic conditions. And these conditions in Patagonia are modern, like from the last probably 1 million years ago. And the record of this Bernardiidae can be traced to the late Cretaceous, like 60 something, maybe 50 million years ago. So the phylogenomic is keep changing, and the relationship among Asteraceae species are always changing, and those sister clades that I show you are now considered monophyletic. For example, this is a Jennifer Mandel on the left tree, and the Santini. It's all in the Nature paper from many people from Kew, and well, they're not exactly the same, but most of the lineages that are native to South America are now considered monophyletics. So they take home messages. The analysis of the fossil record reveals the occurrence of many, mostly Pliocene extinct lineages of the Compositae. Sister lineages originated simultaneously. Extant species are mosaic of ancestral and derived characters, so calling one lineage basal, primitive, and the other advanced might be misleading. Extant Compositae, the light-on branches connecting directly to the root, are not more closely related to the root than any other living species. Okay, thank you so much. These are Viviana Barreda and Cristina Tejeria. They are very shy. They should be presenting this because they work on this subject way more than I do. So thanks to them also. Thank you.
Thank you very much. That was a very interesting talk and good points that we often forget about. So do people have questions for Luis? I'm not sure if that's a question or not.
I have a question, I think. Hey, Luis, thank you so much. I love it. So sometimes, you're trying to describe positions in a tree, right? I mean, and sometimes we say like the base of the family, which even that, right, is sort of misleading. I mean, I wonder if it's better to just talk about tribes and where they're from, or, when you're talking about their positions, do you have any advice for, when you're struggling to describe in a 3,000 terminal tree positions? Do you have any advice for those of us out here?
It's good. Yeah, I mean, we can call them sisters or, yeah, sister families. Sister family is native somewhere, and that's it. And that won't be a lie or a
Right.
misleading concept, or yeah, we cannot say old or young or early divergent, especially if we are talking about living species. If
Right.
we're talking about fossils, yeah, they are ancestral. They are maybe they must or very likely are extinct. But I know it's difficult to find because even myself, I sometimes, well, ancestral, yeah, it's easy because everybody knows what I'm talking about, and
Right. They know what you mean, but it's still
yeah,
misleading. Yeah.
yeah. But I mean, the most accurate term will be, yeah, sisters.
That's true. Cool. Thank you.
I had a question about some of the fossils, and that was, do you have enough detail in like the pollen fossils to know, like, do the fossil pollens have unique characters that are absent in living pollen, or is it just, are there not enough characters there so you can't tell? I don't know if that makes any sense.
Um, yeah, it's, I mean, as I said before, it's difficult when the species are extinct.
Right.
And they might, for example, if we find some Cretaceous pollen grains or Paleocene, which is like roughly 50 million years ago, I mean, it's a long time ago.
Yeah.
And we don't find all the characters. We find some of them, for example, from the pollen wall or some tiny details that we might think they're related to Asteraceae, but it's difficult if you find a capitulum, yeah, it's much easier,
Right.
of course. But pollen grains, you have to know them like in very detail, especially the wall structure, which is very, very typical. I don't know if I answered your question.
Yeah.
Sorry.
Yeah. No, that makes sense. That makes sense. Yeah, it's very, very complicated to find all of this information with just the little hints that you can get.
Yeah, of course, you can always email me, and I'm happy to respond any
Sounds
question.
great. Sounds great. Thank you. So do you have any more questions? Okay, if not, then I will introduce our next speaker, Diego Morales-Briones, who will be presenting their work on exploring hybridization in Flaveria, Asteraceae, the prime model of C4 photosynthesis evolution.
Okay, thank you, Abby, and thank you, Eric, also for the invitation. Just a disclaimer, I agree with Erica that my presentation would take 30 minutes, but I will try to go as fast as I can. And I usually speak too fast anyway, so it should be fine. I will try not to take too much time.
I'm
Okay.
sure it will be good. If it goes over a
Yeah,
little bit,
it
it
should
will
be okay.
be just fine.
Let me share my screen. And you can see my presentation there. To move. Okay. And also, another couple of disclaimers, neither Asteraceae or C4 photosynthesis is my area of expertise. This was more of a side project in the group of the Professor Cartwright, which is mainly interested in C4 photosynthesis. And I think a couple of you have seen this before because this is a published work. We published this in 2023. So I want to start about a very short introduction about C3 and C4 photosynthesis. As you know, most plants carry C3 photosynthesis. This means like the CO2 is fixed by rubisco in the chloroplast, where the photorespiration takes place. And the product of photorespiration are sent to the mitochondria to be recycled to produce CO2 and ammonia. And this can happen both in the mesophyll cells and the bundle shells, in the bundle sheet cells here. And then we have C4 photosynthesis when the main enzyme that's taking CO2 is the Pepsi, and all these products are sent to the bundle sheet to perform the photorespiration. And the main key of this is the Kranz anatomy, when it's like with most of the chloroplasts closer together in the bundle sheet. And there's a lot of intermediates between these two. We're going to call here the C2 photosynthesis, the kind of an umbrella of all these intermediates. And here is just one example where also the CO2 is fixed by Rubisco in the chloroplast of the mesophyll, but then you have a glycine shuttle within the mitochondria of the bundle sheet, where the glycine is recycled to produce the CO2 and the ammonia. And here in the bottom, what I'm showing you here is just the darker green means more presence of chloroplast in the mesophyll. And here in light, it means like the chloroplasts have moved mainly to the dark blue here in the mesophyll, in the bundle sheet, sorry. And that's when the main activity takes place. And C4 photosynthesis has been very important for evolution. There's an estimate that more than 7,500 species can perform C4 and within 20 families. And we have an estimate that at least 65 times has evolved in angiosperms. These are a bit also, maybe there's a little more examples now. And C4 photosynthesis is not only important for evolutionary biology, but also for economic importance. There have been many efforts to trying to convert crops from C3 to C4, especially to try to produce, for example, the C4 rice to increase the yield and also for food safety. So there's a lot of money involved in this too. So now we're going to talk a little bit about Asteraceae, what you like most here in this group. And also I will be talking not only about Flaveria, but another groups later. So, sorry. Flaveria is a group of around 20 species that occurs between southern Mexico, southern US and northern Mexico, all the way to Central America, but mostly diverse in the Sonora Desert. As you can see here, these very nice pictures of the Flaverias. To be honest, I have never seen it in the field. I think I have seen one in Quito, one of the few species that grows in South America. And Flaveria has been the model of C4 evolution for the last 4 years. And this is because within Flaveria, we have all the photosynthetic types that you can imagine. We have C3 all the way to C4 in all the intermediates. Here is not important to see the characteristics table. But for the last 40 years, the scientists have been using Flaveria to explore every single detail about C4 evolution, the morphology, anatomy, chemistry, and everything you can imagine. And based on all these experiments in Flaveria, has been proposed the model of C4 evolution, which again is based on Flaveria. And where you have the three main stages, you have the functional C3, the functional C2, and the functional C4. And there's, as I said before, there's a lot of intermediate within here. And what it changes is like, first you have a full C3, and then you can start acquiring some characteristics that can enable you to develop C2, for example, gene duplications. Then you can start develop the Kranz anatomy. For example, there's some move of the organelles from the bundle sheet, from the mesophyll to the bundle sheet. And then you start having expressing the C4 enzymes at the beginning a little bit weakly, and then you start expressing a little bit more, even more, until you have a phenotype that is functionally C4 that can still have a little bit of C3. This is when the limit here. And in the end, you have a fully optimized C4. So the whole point of showing you here is like the model of C4 evolution in Flaveria is a vertical evolution. It's a linear, it's assuming the vertical evolution. So if you put this in a phylogenetic context, what we expect here is like you have C3 as the sister of the rest, and then you have this kind of gradient here between all the intermediates, and the final species to appear would be like the C4 species. So what has been done in the work with Flaveria in the past, this is the first phylogeny from 2005, when they included 21 of the 23 species, using the traditional markers at the time. And the main found here was like they found clade A and clade B, sorry. And we have been using this nomenclature since then. And the whole point here is like the C3, C4 intermediates of Flaveria appear in two different clades. The clade A is mainly C4 and C4-like, and the clade B is mainly made of the intermediates. So we can say from this tree that the transition from C3 to C4 occurred twice in Flaveria. Then a few years ago, there was the first phototranscriptomic analysis of the group, where they include more than 2,000 genes. They did a concatenated tree. And again, they found the same topology, clade A and clade B. And what they specifically, one of the many things that they did here was specifically to try to see if there is hybridization, because they knew the sample of Flaveria pringlite that included was a greenhouse hybrid. So they developed a test to identify this, and they only concluded the sample of Flaveria pringlite at the moment was hybrid, but no more hybridization from there. Then several years later, they used the same tree to study some gene expression and sequence evolution in Flaveria. And they expanded the test of hybridization that they used in the past to discard completely the origin of the C2 intermediates in the group. Okay. And most recently, at in 2023, they revisit the phylogeny of Flaveria. What was most important here is that they replaced most of the transcriptomes to avoid any confusion from the greenhouse. And although they were focusing on the linearis complex, they again found the clade A and clade B. And all these trees that I've been showing pretty much support this idea of this vertical evolution of the characteristics of C4. So I think it's fair to say that the current model of C4 photosynthesis evolution is clearly the best available that we have. But in the group of first scholarship, we always have been asking, like, what about potential hybridization? But this doesn't come out of the nothing, right? So they wrote a review about this. And the whole point is like, since 1978, when Powell wrote the first systematic analysis of the monograph of Flaveria, he did a lot of hybridization experiments. And at that time, he just took the 16 species that I highlighted here, and he pretty much make artificial hybrids in every possible combination. And he found that most of them can produce variable hybrids. Okay. And since then, and at that time, he didn't know much about the C4 pathways, the photosynthesis pathways anyway. And since then, there has been shown that they have been working in many more hybridization experiments in Flaveria and in other groups. And the main point of this table, if I wanted to take from here, is like, you can take any species of Flaveria and you can mix them, hybridize them, and you can create artificial hybrids that have, like in this case, the AC3 protocans characteristics. Then you can make another combination. You can have the C2 type 1, the type 2, and you can even hybridize some species to create some artificial C4-like. So the whole point here is like the experimental C2 intermediates are indistinguishably, and more importantly, physiologically, from a natural hybrid, so not the natural intermediates. So you can go to the field, find a true species that is a C2 intermediate, and if you can make your own in the lab, and it looks exactly the same characteristics. So that's why this hypothesis of like, if they can hybridize so easily, if you can reproduce this physiology so easily, why not to think that the whole hybrid, the whole evolution of C4 is in some way be facilitated by hybridization. So yeah, so this was the main question here. If it's possible that like Flaveria has been undergone some reticulation history. So for this, we did not create more data. We just took the published available transcriptomes. So we took the 17 transcriptomes available at the time. We added 4 genomes that outgroups, and using the standard methods now, I identified more than 2,000 orthologs that had at least 500 base pairs and include all the species. And again, I just carried some the traditional concatenation and species tree methods, and I explored 4 gene tree discordance. So here I have the the phylogeny of Flaveria that we made. So pretty much the topology is in concordance with the previous ones. There's no surprise in there. We have that the C3 species existed to the rest. We identified the clade A and the clade B. But what we found this time is like this massive discordance within the clade, in the backbone. Here you can see this pretty much cloud. And there's a lot of discordance within the clade A and the clade B as well. And I also carry some coalescence simulations, and the whole point of this is like ILS alone cannot explain all this discordance. So this might be a hint that there's hybridization happening there. Another thing that I want to say here, you can see here this Flaveria pringli in red, the C3. That's the wrong position because this is the hybrid species that was identified before that I included on purpose. Because I wanted to see if the hybridization methods that I use can also detect them as a hybrid. Okay. So regarding the network analysis, we use the same orthogonal that we used before. We use, in this case, Hugo, Maximo, Lilahod, and Fallonet to estimate the networks. And we allow up to 12 reticulations. We collapse the uninformative nodes. And we just plot the pseudo-Lilahod scores to see which is the best model. So first, here is a network analysis with the 17 species of Flaveria that I sampled. So this includes Pringley here as a hybrid. So that's good because we knew that that was an artificial hybrid. So it was a reassuring that these methods work. And we also found the Flaveria brownia was also a hybrid in every single network that we have. And again, the Pringley was known. And the brownia was a surprise for us because we were not expecting that to be a hybrid. But then when we published this as a preprint, Mare Stata and Professor Sage, he contacted us and said, by the way, the sample of brownia that you use and you preprint is actually a mixed sample of 2 DNAs. And that's exactly what we found. So at least we know that those methods work, because I know that we tend to use all these phylogenetic methods of phylogenetic network analysis. And we throw some networks there and we made this crazy hypothesis about hybridization, but we can never check. In this case, we wanted to check, but we didn't want to, but it happened that we checked. So it was reassuring that these methods can detect this. And we remove Pringley and we replace brownia for a new transcriptome, and we carry out this new analysis again. So in the end, the best network was the one with 8 reticulation events. I know this is very complicated, usually these figures, but I wanted to walk you through the main hybridization events. So the first one here in red is showing that the whole clade here, the whole radiation of Flaveria, came from putative hybridization events between the 2 C3 species. Okay. So first we have the 2 C3 species hybridized, and the rest of the species came from there. Then we have that there's a potential hybrid origin for the whole clade A and B. So here we have in green. In both cases, these 2 species, Sonorensis and Angustifolia, which are C2 intermediates, are the parental species of both clades, the clade A and clade B. Okay? And there's other several hybridizations within the clade A and clade B, but those are not important. And this is the original clade B in red. In terms of whole genome duplication, given that there were so many hybridization events, we were interested to see if this might be like allopolyploid events. So here I'm just showing these 3, the number of duplicated genes as a node level. And the whole point here, like we did not detect any whole genome duplication events here in Flaveria. So, and we know that Flaveria is consistent diploid with 18 chromosomes. The haplochromosome number is 18. So, which is surprising because usually homoploid hybridization is quite rare to detect. But I guess maybe that's not that rare in Asteraceae. I guess like the sunflower is another classic example of this. And this is what I have Flaveria, but now because this question goes beyond Flaveria and Asteraceae, it's like what we have seen in other C4 clades. So here there's another word for a group. In this case, this is in the Amaranthaceae family. Here, among other things, we were interested to compare the expression of photosynthetic traits in this group Salsola and this aggregate in the Canary Islands. So what we're interested in this is because this aggregate here in purple is a C2 intermediate. And we have the hypothesis that this group is a hybrid between a C3 and a C4 clade with species. And for this, we also collect some transcriptomes with this similar analysis with the phylogeny that we have here. And not surprisingly, again, we found extensive gene tree discordance in the tribe that has the C2 clade there. And showing you a similar results here, the same methods with pseudo-maximum livelihood. In this case, the networks are a bit easier to interpret. There's 3 reticulations, and purple again is the clade with the intermediates. And in this case, indeed, we found that the Salsola divaricata aggregate is a hybrid of a C4 species, in this case, Salsola soda, and Salsola montana, which is a C3 protocans, which is pretty much a full C3. And we also have the C4 clade is a hybrid of that this C4 clade here in blue is also a hybrid event from a C4 species. And the last one is it's showing us the most members of Salsole are product of an Asian hybridization involving C3 lineages, in this case, Salsole genisinoides, which this matches the same pattern that we saw already in Flaveria. In this case, again, the whole radiation of these clades that include the C4, the C2 intermediates, is from an Asian hybridization of C3 lineages. And we also consider that the C2 photosynthesis and the divaricata aggregate combines traits inherited from its C3 and C4 parental lineages. So we not only detected this in the hybridization network, but also in the analysis for the paper that also identified this. And the last group I want to show you is about, we did similar analysis trying to explore possible hybridization in Echinacea. Here in 2020, they using carbon isotope ratio as Ludwig exchange and C4 pathway biochemistry. They found 2 instances of C4 species in the American xerophilic clade called NAX of the Echinacea. So we have Alionia, and we have this clade of Harbia and of Kenya. So there's 2 instances of C4 photosynthesis in Echinacea. And there's no other C4 species within the family. And together with transcriptomics, they showed that C4 photosynthesis evolved twice in Echinacea, not only based on the tree, but also in other analysis. So again, with this similar analysis I showed you before, we reanalyzed the data, we built the tree, and we explored for reticulation. We found that the American xerophilic clay has full support, as you can see here. But again, no surprise, the backbone has a lot of discordance. And we used hybridization networks again. And we found that this Alionia incarnata, the C4 species, seems to be involving a reticulation with a C4 lineage. But in the case of the clade of Harbia of Kenya, there seems to be a hybridization, but if you check the inheritance probabilities are pretty low. So I would discard this actually. And there's another event of reticulation within 3C lineages again within the clade. So just to conclude, it seems that hybridization might have played a role in C4 evolution in Flaveria, with an initial hybridization event between 2 C3 species. And this might be possible the trigger for the protocan cells in Flaveria. Then we have the C2 species in Flaveria are actually not the result of hybridization between the C3 and C4, but they are in Salsola. And then we have that 2 C2 lineages seems to be involved in the hybrid formation of the clade with full C4 photosynthesis, which indeed support the current model of Flaveria. But instead of being just vertical evolution, we suggest that maybe hybridization has facilitated in some way to acquire all these traits. Hybridization seems to be present only in one instance of the C4 evolution in Echinacea. And most recently, I just read a couple of papers in the last few months. For example, here Pereira Alvarenga recently published a paper in Homolepis, a poesy, when they show again that the C2 intermediates in the case are hybrids of C3 and C4. And they also show that that's why it's important that we take in consideration the hybridization for the model of C4 evolution. But also there's from the same group in other same working group, but in other group of plants, in this case in Acanthaceae, they show there's some recent hybridization in there, but they conclude that C4 primarily arose through vertical evolution, which suggests that the C2 state is essential during C evolution. So claiming that you need the C2 before the C4, which is evidence for the current model. So basically what we have so far is that hybridization is present in all the C4 groups that you can investigate. However, it seems that there's not like a really common pattern. And I think that makes sense because if you saw all these intermediates, C4 photosynthesis or in C4 evolution, is a very complicated trait and have evolved so many times in angiosperm. So it seems like there might be not only one way to acquire this C4 photosynthesis. And maybe in some clades, hybridization might have helped the acquisition of C4 photosynthesis, and maybe not. But I think we need to keep studying other groups for see what patterns we can see. Yeah, if you have any questions, I'll be happy to learn. Thanks.
Thank you for the very interesting talk. That was great. So, well, do people have any questions? I can ask one question if you'd like, and that is just how do you have any idea? I mean, I know you talked about the protocrant's anatomy, but do you have any idea otherwise why hybridization between two C3 species is giving rise to photosynthetic diversity?
Yeah, I think that's the next step.
Mm-hmm.
I think that some analysis of the physiological scale needs to be done. Because this was just in the phylogenetic context. We just have the evidence that two C3 lineages are hybridized, but we don't really know what is actually happening in there for that.
Yeah.
I mean, it's known that the first stage to go to move from C3 to C2 to C4 is to acquire the protocrant's stage. And for that, it's well known that that has been product of genome duplications. So maybe hybridization have helped at the beginning to acquire these duplications and evolve these new functions. And maybe there has been diploidization again, but I think, yeah, we need to keep looking for this.
Interesting. So maybe it's like almost homoploid, but not quite homoploid or something like that. So you're getting duplications. Wow. Yeah, it's so fascinating. Okay, anyone else? Okay, well, thank you, Diego. That was very interesting. So now we have our last speaker, Gonzalo Rivas-Torres, who will present their talk on Scalesia, Asteraceae, Darwin's giant daisies, a model system for studying ecology, biogeography, and evolution in the Galápagos.
Hi, everyone. Can you please confirm you can see my presentation or you're seeing something else?
So we're seeing the presenter view.
Presenter view. Okay, give me 1 sec. Sorry about that. I have
No problem. It's difficult. Get Zoom to do our bidding.
Monitors. These should work. Is that working now?
Yes, perfect. Thank you.
Awesome. Thank you very much. Well, thank you for having me as well. Great to share this space with so great talks and colleagues. So as Abigail was saying, I'm going to be presenting you this group of Asteraceae that live in the Galápagos, and they are called the Darwin's giant Daisies, and how we have been using this group as a model for studying ecology, biogeography, and evolution in the Galápagos Islands, right? I belong to Universidad San Francisco de Quito. I'll also tell you about one field station we have in the Amazon as well, not only in Galápagos. So we know, Galápagos is this particular and unique place, right? It's isolated, it's far from the continent, 1,000 kilometers in front of Ecuador. and it's a young archipelago, right? We know also that this isolation and these unique conditions have been creating also unique, not only organisms, but also unique mechanisms that have been resulting in unique ecosystems. For example, we know that 7 out of the 9 native ecosystems we have mapped for the Galapagos are what we call endemic, meaning they only occur in Galapagos with their particular conditions and particular vegetation. I'll discuss a little bit about this map in a second. And also in Galapagos, 1 or 2 out of 3 native plants is endemic. So endemism is also very high. And among these endemic groups, I'm going to be talking about a very particular one that is Scalesia. So, but before that, it's really important to understand that this, in spite of the very recent descriptions regarding Galapagos native ecosystems, this map I'm presenting you here, and also all the work that we have been doing in Galapagos for decades now, little is known about the ecological patterns, distribution, evolutionary relationships, and conservation of endemic plants in this archipelago. And this lack of key information is also derived through or due to the relatively few collections available that are not properly curated. And we're talking about plants here, right? And you can see here in this map, all the collections, all the dots are all the Galapagos plant records we have had so far, right? All herbarium and research records, etc. So you can say, well, but there are some islands that you have good coverage, right? Like, for example, Floriana and Santa Cruz here in the center. But if you do a zoom in into these islands, you can notice that the pattern holds, right? So most of the collections we have, and therefore the knowledge we have for the Galapagos, are located close to roads. You can see those lines are roads, or main cities and towns, right? But nothing about the rest. So one of these unique taxa in Galapagos, of which we don't know much about the spatial distribution of these species, also its ecology and evolution, etc., it's Scalesia. As I said before, we call them the Darwin's giant daisies. This is because they're literally, as you can see here in one of the pictures here, that is a tree-like form, right? That is Scalesia pedunculata occurring in the highlands of the Santa Cruz Island. And we also have shrubby species like Scalesia finis, you can see here. And you know, out of the 7 endemic genera of the Galapagos, this is one that caught Darwin's attention. Okay, this is really important to remember because Darwin was there, of course, as we know, but he was particularly interested in plants, and he collected several of these specimens of Scalesia, and then sent it to some botanists and colleagues, and therefore some new descriptions happened, right? Therefore, you know, because we don't know much about this group, one of the general objectives of my laboratory, and in collaboration with several colleagues, you will see that in a second, we have been working on this group for over a decade now. And despite, you know, this genus has 15 described species, as I said before, many of them described after Darwin's collections, and is recorded widespread in 11 islands of the 13 that we call big. And because of this, you know, particular distribution, we call them also the Galapagos finches in plants, right? It's like you have in island endemics, some of these unique taxa only occurring in one island. And as I said before, and as I presented some of the types there, there are several species that have been catching the attention of many people, including Darwin, that collected these Scalesia incisa in 1835. And then after this, we don't know much more about these species, right? So it was the description and that's it. We don't know much about the ecology, about the evolution of them, et cetera, et cetera. So that's why we have very few studies describing the Scalesia biogeographic or biogeography, ecological and evolutionary patterns at relevant scales. What I mean by relevant scales, that can facilitate the generation of hypotheses, for example, microecology and related fields as well, enabling, of course, informed conservation and decision making, really important. So before the maps I will present you in a second and some of the results I will show, this was the best map we have for the distribution of Scalesia, right? And this was a fantastic job done by Ito and others until the mid 90s and late 90s. But of course, we need more detail, right? As I said before, for not only understanding better the ecological and evolutionary patterns of this group, but mainly to know how to do proper management and conservation for this. So today I'll show you really fast three important results or objectives of the research I have been doing on this group. First, describe the distribution and endemism patterns of Scalesia, provide insights regarding the diversification of this group, and of course, genomic and evolutionary patterns for this endemic group, and present the most up-to-date information on Scalesia ecology and some abiotics or biotic interactions. So in order to improve the number of collections we have for Scalesia, right, I was all over the archipelago. I'm very lucky to have been traveling around these islands for more than 10 years. And every dot you see there are plots that I have been establishing around populations of Scalesia. I have also been able to fly drones on top of these populations. And as you may notice here in these green dots, these are Scalesias that are evergreen. So in the dry forest where most of them occur, it's pretty easy to literally geotag individuals, right? These kind of methods have allowed us to include or to add up on average around 4,000 individual records for all the 15 species. So now we have enough information to create things like this, right? Range sizes or areas of distribution that using these collections I show you and modeled by ecosystem and biotic conditions can tell us exactly where these populations are distributed. And as you can see here, some of them are in volcanic tops, like Scalesia microcephala, in the volcanic tops of Floriana and Isabella, the Big Island. With that information now, for the first time, we have detailed maps on confirmed and curated Scalesia locations, as this is extremely important, right, for some of the results I'll show you later. And of course, we have also maps showing the range size using this ecological approach that can help us to also monitor these species that is key for this archipelago. We are also collecting other information derived from this, right? Basically, for all the 15 species you have there, how many of them are in which islands, right? Not only where, but how many islands are these island endemics? Are these occurring in other places? And as you can see, 12 or 15 of these species are occurring only in one or two islands. So they are very restricted. The other thing we can start understanding is that we have a species like Scalesia finis in the top square there that can be distributed in thousands, literally thousands of hectares. And you have others like Scalesia aspera at the very bottom that are only occurring in like a few hectares in the archipelago. We now understand also that the tree-like species are occurring mainly in lowland to highland, so they have a widespread in the archipelago, and most of the shrub species are occurring in the lowlands. As a second objective of the talk, and really fast, as I told you before, not only by establishing plots and doing drones, I was able to collect ecological information, I was also able to collect plant tissue. And with that information in 2020, with colleagues from Jardín Botánico de Madrid and others, we put together the first phylogeny of Scalesia that you can see here, right? So basically, each color belongs to each island, each color in the clade. And what you can notice are several important things about this phylogenetic tree. One is like there was only one introduction, like maybe 3 million years ago or so, of one ancestral species that arrived to the island and then gave rise to all these 15 species. The other thing you may notice is that, and if you know something about this archipelago, the islands here located at the east are older than the ones located at the west. And if you notice these two or three species here, these two clades are occurring in the oldest islands of San Cristobal. So we can now know that Scalesia is not really following this progression rule where older, right, taxa is usually occurring also in older islands. And finally, something you may notice is that the leaf shape of Scalesia can vary along the whole phylogeny, and that some of these leaf shapes, like this very lobe shape, is occurring independently in the phylogenetic tree. Since that study, now we have been able to put together other kind of research, including this, adaptive radiation, genomic studies, photosynthetic strategies of this group, and also how microbiomes are assembled, right? And how they are fixed or not through the whole phylogeny. Now, finally, to talk about the ecology of this group, really important or really fast is to let you know that when we started studying this group, there were some papers showing that in the central island of Santa Cruz that you can see here on the left, like around 40 or 50 years ago, we only have 3 different types of ecosystems, right? In the different 2 greens and this kind of like light orange color. But then Truman and others went back to the same plots you can see in that graph in those dots and put and recensus those same plots, and they found out that now we have 13 novel vegetation states, most of them dominated by invasive species. And in islands like Santa Cruz, we only have 1% of the Scalesia forest left. So of course, there are a lot of changes occurring where humans are and the Scalesias are also there. So in 2014, I put together a group of plots that you can see here. Let me see if I can use the laser pointer, so it might be better. So here in what we call the Cedrela block, so this is dominated by the invasive tree Cedrela dorata and the different plots here in the different dots. And we also established plots in the Scalesia dominated forest where the Scalesia pedunculata species dominate. And we measured tons of things, right? Including canopy openness, forest height, leaf litter, DVH, etc. And what you can notice here is that the Scalesia forest plots, the ones in white, right? When we compare all these abiotic conditions with the Cedrela invasive dominated forest, they are significantly different, right? So there are changes in the abiotic conditions. Now, if you follow me on the same kind of like color coding, Scalesia and Cedrela, but then we only look to the native species here where I'm pointing out, we can notice that the abundance, richness, and diversity of native species is significantly higher where Scalesia is dominating. But when we look at the Cedrela forest dominated by the invasive species, then we have the reverted pattern, right? So sure, conditions are changing, and this is what some people is calling a novel ecosystem. So then in 2020, and we just published there, I went out and started cutting down Cedrela, planting back Scalesia, do a restoration project basically, and sowing or planting 4 different invasive species here in this green, in this gray block, and then and also endemic species in the white block, including Scalesia pedunculata. And we follow the fate of this for a year, and we follow seedling emergence, establishment, and growth. And what you can notice here, if you look only at the Scalesia, is that particularly for establishment, right, when Cedrela is present and caught, there is negatively affected the establishment of these species, but when it's not present, then is positively affected according to these models. Same thing we found in growth. So there's a direct competition with Scalesia, and literally this invasive tree, Cedrela dorata, is outcompeting Scalesia. So as conclusions, I'm close to finish. Our research for over more than a decade have been able to allow us to determine that for the first time, the distributions and biogeographical patterns of these Scalesia species that maybe arguably is the most iconic genus of plants in Galápagos. Now we know their phylogenetic relationships and evolutionary processes that are happening in this group. That is really important because, as you can see, it's kind of a textbook example for this kind of knowledge. And also we know that the biotic and abiotic interactions of some of the Scalesia species, as well as the threats to their survival are caused by exotic species, right? And this is providing information for the correct management of these species. So overall, with all these results, we can say that now we can determine that this group is undoubtedly an exemplary case of illustrate ecological and evolutionary processes, particularly in plants and in plants occurring in archipelagos, and the importance of Galápagos as a natural laboratory, also as Darwin evidence, right? So we hope to find more allies to continue this kind of studies. So please feel free to contact me if you want to go to Galápagos, why not? And then do some more research with us in this, just let me know. And with that, I want to acknowledge all my students. Of course, they are the driving force of this. Tons of collaborators, the Galápagos Science Center, one of our field stations in Galápagos, Galápagos National Park Rangers, and of course, other colleagues here. And before I'm finished, really fast, I also want to tell you that we have another field station that is the Tiputini Biodiversity Station in the Amazon. That's the field station I direct currently, sorry. And so you can, if you are also interested in going to the Amazon, we have some Asteraceae there. Solanum altissimum. We have some trees there. So if you want to go there, you're more than welcome to visit us, okay? With that, I'll leave you if you have any questions. Thank you very much for the attention.
Thank you very much, Gonzalo. Another really great talk. So do people have any questions? I can ask a question. One question I had is how much of, obviously invasive species are a really big problem, but like how much of the flora, like by biomass or whatever, is invasive in general? And does it vary from island to island?
It varies. I don't think we know by biomass. Maybe with the new maps we're putting together, we can know that. But just to give you an idea, I think the most updated species list we have from the 890, maybe it's more now, plants we have in Galápagos, close to 500. Let me tell the numbers straight. Sorry, I'm in Germany and it's very late here, so my brain is not going. Don't worry. So yeah, these are the numbers. So we have 500 and so native species and 880 or so non-native species.
Wow.
So it's almost double the number of non-native species now when you compare it to the native species. That's how critical is the problem now. I think there's a new list via the Charles Darwin Foundation, and I think the non-native species number is even higher now for plants. This is only plants. So, you know, and then you have these species I'm showing you, right? Cedrela is it's a tree that have been, you know, growing for millions of years with thousands of other trees surrounding, great competitor, putting chemicals in the soil to outcompete other species. And you introduce that to an island where you don't have really tree species. We have, I think, maybe 20 something tree species in Galápagos. Then of course, this is the perfect space for invasive species to thrive and outcompete these, you know, unique species.
Is there any funding for trying to control these with all of like the ecotourism and everything like that? Or is it?
There is funding. There's a fund through the Galapagos National Park. It's highly competitive. You know, as usual, is not enough. And there's now lots of funding actually for restoration of ecosystems and mainly to bring back some species. Like there's an iconic project in the Floriana Island, the southernmost island you can see there in the map, where they are trying to get back many bird species and Rewild and many other NGOs are helping. But I think, you know, sometimes I'm afraid there's not enough time for like, you know, doing more research. It's like, okay, let's try to protect whatever we can because again, these invasive species are, you know, year to year taking so much land that, you know, we should be doing something. But unluckily, I don't think there's enough.
Yeah.
I don't know, any other questions?
I guess if anyone has questions for any of our speakers. Okay, well, if not, thank you all three for three really interesting talks. A lot to think about there. And I hope to see everyone back in December for our last TICA talks of the year.
Thank you. Thank you for having us.
Thank you.